This is a special episode of the Immunology Podcast, IUIS 2023, Day 1. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. As you all know, yesterday launched the start of IUIS 2023, the International Congress of Immunology, in Cape Town, South Africa, and Brenda, myself, and the Immunology Podcast team are all attending. Drop by the Immunology Podcast booth, number 6, in the exhibit hall to play some games, win a prize, and find out how you could be featured on an episode of the podcast. Today and every day throughout the meeting, we will be releasing special episodes discussing our favorite sessions of the previous day. So if you weren't able to attend, we've got you covered. We're going to kick off things here in just a moment, but before we get to that...
Are you looking for smarter and more efficient ways to isolate cells? Well, EasySep cell separation technology combines the specificity of monoclonal antibodies with the simplicity of a column-free magnetic system, enabling the isolation of highly purified cells from a variety of species and sample sources in as little as 8 minutes. Explore at www.stemcell.com/EasySep to discover how you can use EasySep to advance your research and explore our range of EasySep kits.
All right, well, we got through the first day here, Brenda.
Woohoo, that was fun. Well, 1.5. Technically, there was some events yesterday, but it was only the kickstart of the conference. It's good to be in South Africa.
It is.
Yeah.
It's Monday, right?
It's Tuesday, my friend.
It's Tuesday. That's right. It's Tuesday. Okay. Tuesday, yes. Yeah. Monday was yesterday.
Monday was yesterday. You're getting it.
I'm getting there.
Yeah. So... very exciting. Why don't we just jump into, I just want to say, yesterday was the opening ceremony. I thought it was very nice. It was some music, but there also was an opening kind of speech from the president of the IUIS, also a previous president. And I have to say that was an all-African ensemble that were presenting some keynote lectures also. But I, coming also, from the global south, I thought it was really nice. I think it's great that the conference is here in South Africa. And clearly the organizers were also thrilled, and they really made that point of this kind of historic location. What do you think?
I thought seeing the history of it and the commitment to being here, apparently for the first time, in Africa, it was really neat to see just kind of the phase shift in what's going on. And so it was really cool to see that.
Yeah.
All right. Well, getting in to it, what session did you go to first?
Well, this morning we all went to the keynote by Emmanuelle Charpentier, Nobel Prize winner, who says she's not an immunologist, but after all, isn't CRISPR a immune system for bacteria? So I would say, that is a partially true, partially false sentence. But she, of course, talked about her important her discoveries in CRISPR and the role her team played. But what she ended mentioning, I just want to, for those gene editing fans out there, I think it was last week or very recently, the UK approved one of CRISPR Therapeutics, which is this company that Emmanuelle Charpentier co-founded. To harness the power of her discovery. They actually they have a gene editing therapeutic for sickle cell anemia, and they actually got it approved in the UK. So I guess other approvals probably will be coming in other countries. I think this is a huge milestone in genetic engineering, and I'm pretty sure there's going to be other also for more immunological diseases. Yeah. Big thing.
My wife works for a gene therapy company as a toxicologist. It's moving fast.
Yep. But then we had sessions. So where did you go afterwards?
So I went to animal models of human immune disease.
All right.
It was very pork focused, a lot of pig going on.
Not very kosher, was it?
No, not kosher at all. But no, it was interesting. Not, I think, a lot of insights to share necessarily here. It's a good large animal model, as people know. There's been a lot more work recently interesting. The one thing that came up was the microbiota was really, and specific bacteria are predicting vaccine responses to things like influenza. And so it seems like the host milieu, unsurprisingly, some of that seems, just like we've been talking about in people, also true in swine, which is interesting and thus useful as an animal model. But that was really the only thing I was able to pick up from that first one. How about you?
Well, my first session was TB or not. immunity to bacteria. And I thought I really liked it. I mean, I don't have to say that tuberculosis is still a huge burden all over the place. So it was really nice to see some of these talks. The first session was Joan Flynn, and she showed some results on macaque studies, trying to study factors of protectum tuberculosis, particularly kind of adult onset of tuberculosis. And I thought it was really cool because she showed that although we usually, the BCG vaccine, that it's kind of the only thing we have against tuberculosis for boosting immunity, it doesn't work very well for adult, but so protects to some extent, and usually is administered intradermal. But in this macaque study, she actually showed that intravenous was much more protective than the other aerosol or intradermal. And she did some characterizations on the importance of CD4s or CD8s, and she showed that the T cells really eliminating T cells from vaccinated macaques really increases dissemination. And she characterized some of these responses when you do IV BCG, and also if you have a more than one exposure. So she looked into also what happens when you have a macaque and you infect the macaque is infected, and then what happens if you actually treat. That treatment actually reduces the protection for a second infection. And that's what I thought was cool, is that they showed a different strain of BCG, because IV BCG sounds like a bad idea, like you're gonna inject, technically BCG is a live bacteria, but they have this doxycycline dependent strains. So you don't only if you administer the BCG with doxycycline, that the bacteria is still alive, but as soon as you remove doxycycline, then the bacteria will die.
But you still have to administer doxycycline to people, which is antibiotic and has its own.
Yeah.
You just blasted.
Interesting idea, maybe a different molecule, but I think the idea is kind of cool. Then Emily Wong from the Africa Health Research Institute talked about a study called Bakuzazi, which apparently means wake up and know ourselves. And they screened huge amount of people, 18,000 people for TB. And they showed that actually like 80% of the TB positive patients, they did sputum analyses and they were tested positive, were asymptomatic. So there's all these people walking around, they have TB, they don't know it. And she studied a little bit what is the spectrum of subclinical TB, and always with the idea of trying to pick it up better. What things we can do to pick up these patients. Then Sarah Suleiman, she was talking, she did a GWAS. She was showing some results from her GWAS studies, trying to comparing individuals who were exposed to TB but did not develop the disease, and trying to find the variants that associate with this disease. Turns out it's very hard. Then she combined it with other kinds of studies. She actually picked up some interesting correlations. And I thought was also really cool was last but not least Laura Pallett from UCL, University College London. And she was like, I'm sorry, my talk is not about TB. But she had this really cool story about CD8 T cells that resident in liver, that they are picking up CD14 from myeloid cells around them, and this actually modifies their phenotype in a very interesting way. So they are snatching CD14 from their companions. Pretty cool.
Okay.
Yeah.
What does that do?
So it shows that these T cells, they actually they have less inhibition by PD-1. They seem to respond stronger to TCR stimulation, and they actually can recognize bacteria LPS, and we can be activated in an absence of TCR stimulation. And they do this because of these synapses, and then I think by now it's pretty well known that T cells, they acquire surface molecules from cells they synapse with. And they are and they found first in liver, but then in other places as well.
Interesting.
Yeah.
All right. Well, my next one was on the future of scientific publication, and
oh boy.
Oh yeah, you had some science people there, some nature people there, and some frontier people there. And I'll just say that the conversation was spicy.
Can imagine.
A lot of healthy debate about open access and the costs versus what the journals provide behind paywalls and the benefits of that, and what about preprints and peer review, and nothing was solved, but lots of feelings were shared. And I'm gonna leave it at that.
Oh my. Okay, let me go something much less spicy and controversial. Helminths and antifungal immunity. I mean, you gotta get rid of
helminths
those.
can be pretty spicy.
Yeah.
They can be a little uncomfortable here, folks.
Well, just quickly, I had a talk by Benjamin Dewalls. He was showing that there's some T cells that this kind of self-reactive memory, virtual memory T cells that they're found. They can actually respond to IL-4 and upregulate the expression of CD22, and that this kind of gives these cells provides these cells with some particular properties. So in general, so they express more CD39. They have increased self-reactivity, and they express also effector molecules. And this is I think this has to do with helminths because it's in response to IL-4. Then I also had Suprabhat Mukherjee, who was talking about some surface markers from, I think they're nematodes that cause elephantiasis, and how they have some surface sheath proteins that are recognized in TLF4, and that they activate the immune system in particular ways, and they are partly responsible for the lymphedema that generates the limb swelling or characteristic of the disease. And she suggests that because this is an important protein, that you can actually try to vaccinate against it or inhibit their function in a particular way, and that might actually alleviate the disease. Then I had Mei-Quinn Shi talking about cryptococci neoformans infection in the brain, so aggressive fungal infection, and show that there's this population of Ly6C high mononuclear phagocytes that are kind of responsible for clearing out the infection, and that their recruitment requires TNF alpha in the brain, expression in the brain, and that their function actually is dependent on interferon gamma expression, and that in this also is dependent on CCR2. And she shows it by doing CCR2 knockouts and showing that this prevents reduction of CFS of colony-forming units in the brain of these fungals. And last, Robert Mugo from the Free University in Berlin. And I think it's really cool. He works over Ascaris infection, also a nematode that is on the one hand important for farming. It infects pigs and can be very detrimental, but also can generate chronic infections in humans. So he was looking into ways of measuring the burden of Ascaris, and he compared different things. Like there's, I think, what is the gold standard probably does make microscopy. This is not very high throughput. He compared using qPCR, and he also measured humoral responses, whether you can use antibodies in blood to correlate with infection. And he actually found some quite cool things. You can actually use qPCR to estimate parasite load, but then and you can use certain IgG, particularly IgG1, against certain antigens that correlate very well with positivity measured by conventional things. So I think it's, of course, very important in case of, in endemic areas when you need to do a lot of these tests. Whatever is higher throughput is always good. So really, I had to admit, that was not my first choice of session. I was not allowed in the one I wanted to go, but I'm happy because, sometimes I tell myself, you need to get out of your comfort zone. You can't always go to check out the T cells. You need to see what is there out in the immunological field.
Well, I checked out T cells, Brenda.
You did?
Gamma delta T cells.
Oh, those T cells. What do you have?
All right. So we had 3 talks that all kind of weave together. One was by Yi-Shi Jian. Another one was by Adrian Hayday, and a third one by Emo Prins. And they all kind of tell the combined story. So I'm just going to talk a little bit about the theme, which is gamma delta T cells have weird T cell receptors, which can be restricted to peptides versus very specific epitopes. And so there's some mapping of how some of these peptides or specific marker peptides also had heterogeneous ligand binding. Something that's a Cy3 specific TCR actually binds a bunch of crap. But then the question is, do you need the TCR for their function or not? And it's been very controversial in the field. I'm not a gamma delta person. I've been at gamma delta adjacent a bit, and so playing catch up here a little. But high level, one person had showed that IL-17 responses in some of these cells really require TCR and cytokine receptor engagement. But then they also show that at baseline, you don't need a TCR for them to exist, but you do need them for function. And what they found was that they need co-stimulators, though. And normally, there's a normality sensing component to them. So they can, at baseline, have normality sensing. So like, oh, yourself all the time. That's part of what the TCRs actually do. And that there's these specific mutations and SNPs they've found with mutant alleles linked to IBD where it loses normality sensing. And then they see that that's associated with inflammatory bowel disease. And also just even in healthy people who don't have the disease yet, they have higher levels of inflammatory genes and such, showing that they're predisposed to inflammation. There's also, because of the specifics of how the receptor recombination works, you can do where you can make a specific knockout of the TCR only of gamma delta ones. And so they did that and then did it tissue specific and showed that if you knock it out, you have more chromatin accessibility, and they start looking and behaving more like innate lymphoid cells. And then the last person was really showing some more of the mapping of the repertoires and how these different like kind of clustered repertoires affect the function, because it's not like there's just one single antigen. It's like these clustered effects. So that was that talk.
All right. What else?
What else did I get? I got into some cancer immunotherapy, Brenda.
Oh, wait, but before that, while you were, playing around with gamma delta T cells, I was meeting la creme de la creme. Was at a meet and greet with doctors Emmanuel Charpentier and James Allison, and organized by the IUIS junior community. So thank you very much for organizing this, guys. We had a kind of close quarters meeting with them. And we just had, we got to talk to ask them some questions. So I just want to share some snippets of our conversation with Dr. Allison and Dr. Charpentier. So both of them, of course, the questions, there were some kind of generic questions that were asked by the committee. What brought you here to science? And of course, curiosity was heavily cited by both. I like that in the case of James Allison, he also mentioned a family history of cancer that it really motivated him to work on this field. And in the case of Emmanuelle Charpentier, she already knew from a kid. Her oldest sister was in university when she was very young already, and that kind of already gave her a path. It's like, I want to do what she does, and I want to learn, and then teach, and then be part of this. So I thought that was pretty cool. And I guess also another thing that I liked from Emmanuelle Charpentier, talking about distilled learnings. I think one of the things she said, and I cannot agree more, is the importance of being self-reflective, evaluating yourself, and sometimes ignoring what other people say. I think both of them had their situations. James Allison mentioned how hard it was to convince people of doing a clinical trial. For those who don't know, he brought to the clinic the use of anti-CTLA-4 antibodies for melanoma. These ipilimumab was the first commercial production, and people, he was very clear that people were very skeptical, and he had a hard time convincing them to treat patients with this. But in the end, it turned out to be a game changer. And in the case of Emmanuelle, of course, she moved a lot, so she worked in many, many institutions, and she kind of said that she got bored a little bit. So she had a plan, how long he was going to stay here, and when things she was not convinced, she would move on. I thought it was a kind of a kick-ass attitude. And it's just you got to take the chance and risk to follow what you think is good. So listen to what your gut tells you. There's also words from Emmanuelle Charpentier. And I thought it was really good. It was a nice conversation. So thanks again to the junior community. I hope to be talking about them a little bit more in the next episodes.
All right. Well, I am not junior. We've established that I'm old, apparently. So I didn't get to do any of those cool things. But I did go to a cancer immunotherapy talk. A lot of this was some more of a rehash of some greatest hits of cancer immunotherapy work. And so I'm just going to focus on a couple couple things. One was from Nina Bardwaj about how they're now going after frame shift antigens for vaccines, including microsatellite instability ones, notably things that mimic Lynch syndrome, which is where you have a lot of that's the familial disease with a lot of polyps in the colon or in the small bowel as well. And the idea here is that unlike personalized vaccines for immunotherapy, adjuvants, these can be commercially produced because it's a common mutation. Mm-hmm. And so it's more commercially viable. So that was one little point. And then Tak Mok, who's done a bunch of work before, was really putting out a new case about a high acetylcholine. And there's some trials coming out, and it's not all out yet. But the one thing that I'll share that was pretty cool is that they showed that there's this role of choline, acetylcholine, and it dampens PD-1 and CD25, so Tregs. So acetylcholine is pro-killing tumors. And they showed that in hepatocellular cancer, patients live 500 days longer if they're in the high acetylcholine group and have better responses to immunotherapy.
Okay.
500 days is quite a bit.
Mm-hmm.
So that's one of the areas to look at. And then he's doing a lot of work looking at using metabolite disruptions to kind of have a phase shift in solid tumors. As we know, immunotherapies aren't particularly successful lately outside of liquid tumors. And melanoma is kind of a liquid tumor. Melanoma is considered a liquid tumor.
Well, but there's been some, I would say, colorectal cancer.
Only microsatellite instability high, so which is very rare. So it's like in most of it, you don't have huge responses in a lot of solid tumors. And so what they're finding is, just on its own, it's not working. So is there either biomarkers of metabolism, or are there metabolic pathways that they can go after with small molecules that will then potentiate the effective immune response, because there's this tumor microenvironment that seems to be metabolically sustaining them even in the face of immune therapy. Mm-hmm. And so can you disrupt that? And I think that's the difference is liquid tumors, because they're so disseminated, don't have that microenvironment in the same way, right? Your blood is your entire bloodstream. Melanoma is pretty liquid and disseminates everywhere. It's kind of a unique cancer, and so that's what they're really pushing on, and so they're hoping that this is how they can get to some solid tumors.
I just want to say that before the meet and greet with Manuel Charpentier and James Addison had also gave a keynote lecture, which was also very interesting, and I think he also made the point of sometimes combining immunotherapies and combining approaches can be very important. So the last session of the day I attended was Systems Immunology. It was chaired by Mark Davis, former guest of the show. And he really is into, I think lately he's been pivoting a lot towards try to compare or look into human immunology, because mouse is always hard to translate. And he has been developing using organoids, and particularly those derived from tonsils and apparently also from spleen. And so these kind of self-assembly single cell organoids, apparently you can reassemble them from single cell. And he has used them to study, on the one hand, influenza vaccination, looking around the idea of the original antigenic sin, this idea that the first influenza you are subjected to might kind of condition the other influenza's responses. And he showed there were some studies using twins that suggest that actually it's not so much the initial flu exposure, but it's more than the genetics and the HLA presentation and the HLA repertoire that you have and how good it is at presenting certain peptides over others. So he used this tonsil organoid system to compare different vaccine approaches. So showing that cross-linking antigens from different hemagglutinin subtypes in one molecule actually prevented this disparity in the immune response, because she showed that it's better to have all the antigens in one big ball, so to say, than having individual vaccinations with all the antigens, because by themselves some antigens are better at kind of being presented and activating T cells. But if you have these dominant antigens on the same dendritic cell, that dendritic cell would get CD4 help and then be better at presenting all of the antigens, including those that are weaker. And so this actually can give you a much more balanced immune response, also against antigens that you would be kind of predisposed to be worse at presenting and at generating immune responses against. He was also looking into tonsil organoids for studying CD8 suppressive T cells that are expressed in mice. They express Li49, and I don't remember what was the marker for in humans. But these are also CD8 T cells that actually kind of have an overall suppressive function. And he also showed that, tonsil organoids and ginkgo cultures to study rejection in vitro. And the fact that now they're doing organoids from human spleens, I thought it was pretty cool. So I really like that. I need to look closer into that, because this whole idea that I think what he's trying to say is, how can we study the human? How can we generate models for studying more properly the human system instead of having to only rely on mouse? And then we have Sarah Teichmann from Wellcome Sanger Institute, who is apparently on a quest to make an atlas of every kind of tissue and its immune cells. So he has some very, very sophisticated models for kind of modeling space and time. And he was particularly showing some data on thymus. He got a hand on a lot of thymi from different ages, from kind of really, I think it was prenatal and kind of infant thymus. And she used these with some very fancy measures, spatial transcriptomics, to make a model or to kind of generate a model of thymus and thymus development, which was pretty cool. And her lab does this with many other organs. So she did seem to be on a mission here. Very, very, very impressive. And last, I really like the doc from Peter Brodin, that he's at Karolinska and also I think somewhere in London. Well, he's looking into different sex differences in human immune responses. Of course, we all know, and he showed some data I thought was really cool from COVID responses, how males did pretty, in general, worse than females. And this, in principle, was characterized by reduced interferon type 1 response that then, in the end, it ended up being a more stronger inflammatory response with IL-6 and more kind of damaging response.
Are you saying the man flu is yet again, established as true?
Bah, no, yeah.
Worse.
You weak man. Man
flu.
Yeah, I guess that's that's the interpretation you could do. So what he did, so it's always hard. What is genetics? What is hormones, right? And so what he did, he studied people transitioning, doing gender affirmation care with testosterone treatment. Huh. So I thought that was brilliant. Then you have
and the chromosomes won out.
The chromosomes, so you don't have to. Those don't change.
Yep.
So she showed that after 1 year of gender affirming testosterone therapy, these patients, they show kind of this shift from towards a decrease in the type 1 interferon response towards an increase in the TNF alpha and inflammatory cytokine responses. And he showed some subsets, for example, PDCs, which had an increase in CD81, and this correlated with the less production of interferon type 1. So I thought it was really cool. I mean, a really good, smart system to study this.
No, that's neat. So it is hormone driven then.
Yeah. And he also showed that this, his talk was longer than this, but he also showed that this also changes in the female cycles. You see differences in this. And he hypothesizes that this increase in interferon response and decrease in inflammatory response might be a preparation for gestation. So you want a woman that has a fetus to not be so inflammatory, but still keep their antiviral immunity.
You have to be able to tolerate the parasite, right?
Yeah, indeed. So I thought that was really cool. I really liked that last talk. So we went out on a good note. It was a great day.
That is it. Well, that brings us to the end of our first IUIS 2023 episode. Don't forget to follow us on Twitter at @ImmunoPodcast to find out what we're up to at the meeting, and visit us at the Immunology Podcast booth on the exhibitor floor where you can win a gift basket. Check back here tomorrow for another episode recapping day 2 of the meeting. See you then.